Great. Thank you so much, Dr. Wang. I think we're out of time, so I'm going to pass the mic to Dr. Salim to introduce our next speaker.
Thank you, Dr. Nata.
Thank you.
Now we have our next speaker, and I have the honor of introducing Professor Meri K, who is a professor and research scientist at the Mediterranean Center for Molecular Research at the University of Nice. She's going to deliver a lecture on analysis of matched skin and gut microbiome of vitiligo patients reveals dysbiosis in lesional skin. Over to you, Dr. Meri.
Good afternoon. My name is Meri Tulic. I work with Professor Passeron in Nice, France. I'd like to thank the organizing committee for giving us an opportunity to present our work here today looking at dysbiosis in lesional vitiligo skin. Vitiligo is an autoimmune disease characterized by skin depigmentation due to the loss of melanocytes. Although VIS studies have shown more than 50 genes to be associated with vitiligo, the delay in age of onset over the last 30 years emphasizes the role of environmental factors. We have recently shown that innate immune cells are more sensitive to stress signals, and their activation is the initial trigger in melanocyte destruction. Bacteria are among the top producers of stress signals. There is now a large body of literature showing gut dysbiosis to be associated with skin disease, and more recently, skin dysbiosis to be associated with skin disease such as atopic dermatitis, acne, and psoriasis. Here we set out to examine the role of skin microbiota in vitiligo. To date, only one paper exists in the literature by Ganju and colleagues who have looked at surface microbiota in vitiligo skin without comparing it to healthy skin, showing only small changes between lesional and non-lesional areas. A more recent study in a mouse model of vitiligo has shown that depletion of Bacteroides in the gut may induce skin depigmentation, suggesting a possible link between gut and skin. In our study, we recruited both vitiligo and healthy controls. We took surface swabs, but also deep skin biopsies and gut stool samples, sampling different sites. We also took skin samples from both lesional and non-lesional sites, sampling different zones. And we also collected blood samples to be able to examine changes in microbiota with immune function. Initially, looking at gut microbiota, this slide represents different measures of alpha diversity within the sample, looking at the number of species, the diversity, and richness. Results have shown reduced alpha diversity in vitiligo samples compared to healthy controls, demonstrating for the first time changes in the gut of vitiligo patients with skin disease. Here we see composition and diversity of our samples by comparing relative abundance of bacteria on phylum level in 10 control and 10 vitiligo patients. We see a trend for increased Firmicutes in purple and decreased Bacteroides in green, resulting in significantly increased FB ratio between the two groups. In the literature, increased FB ratio significantly and strongly correlates with obesity, cardiovascular disease, as well as autoimmune disease. And interestingly, it's been shown that Bacteroides is protective in the gut of mouse vitiligo model, hence its loss may contribute to onset or exacerbation of the disease. To examine if there are differences between swab and biopsy samples from the same patients, we measured alpha and beta diversity in each of the paired samples and have shown that both number and diversity of bacteria were higher in swabs compared to biopsies. And we also saw a significant difference in beta diversity between biopsies and swabs, which allows us to conclude that microbial diversity and richness differs in relation to sampling method. The most obvious difference is that while in the swabs more than 30% of genus belongs to Staphylococcus, while in the biopsies Bifidobacterium makes up almost 60% of total genus, suggesting that certain genera correlate strongly with collection method and that there is a need for simultaneous sampling of both surface and deeper skin layers to get an accurate picture of changes in the skin microbiota, which has important implications for interpretation results. So how did these results differ? Initially looking at the swab data, although results have shown increased alpha diversity between control and vitiligo samples, we saw no significant changes between non-lesional and lesional regions. Examining the composition of these swab samples, first we see significantly decreased firmicutes in orange in lesional compared to non-lesional sites, most likely due to decreased staphylococcus in blue at the same site. We've also seen trend for increased proteobacteria in green in lesional compared to non-lesional site, which is the bacteria containing many known human pathogens associated with skin inflammation. Moving on to the skin biopsies, in contrast to the swabs, we now see differences in alpha diversity not only between healthy and vitiligo biopsies, but between non-lesional and lesional vitiligo samples. In lesional samples, we saw decreased species diversity, but increased community richness compared to non-lesional sites. Importantly, in contrast to the swab microbiota, we've demonstrated that the lesional biopsies in red had the most distinct microbiota compared to all the other groups. The increased richness can be seen in these taxa plots, showing lesional biopsies to have decreased actinobacteria in orange and increased firmicutes in purple. However, the largest and the most significant difference is the presence of tenoricutes in pink and mitochondrial DNA in brown, exclusively in 7 out of the 10 lesional sites. This is interesting as mitochondria has been implicated in ROS production in vitiligo cells, and in mice, high mitochondrial DNA has been linked to reduced microbiome diversity. Looking at the genus taxa plots, the most prominent observation is the almost complete absence of bifidobacterium in 7 out of the 10 lesional sites. In lesional biopsies, in general, we saw a depletion of commensals such as bifidobacterium, E. shigella, and staphylococcus, while there was an enrichment in pathogenic bacteria such as mycoplasma and streptococcus. This was seen in the same 7 patients who had strong increase in mitochondrial DNA. To try and understand why these 3 patients behave differently than the rest, we looked at their immune function because there's a presence of bacterial DNA has been linked to the loss of bifidobacterium, which we know is an important protective commensal shown to decrease the activation of innate immune cells. We measured serum levels of innate and stress markers and have shown that 7 patients with increased mitochondrial DNA in lesional skin had significantly increased immune function compared to patients without elevated mitochondrial DNA, suggesting that mitochondrial damage is associated with increased immunity in the same patients. So in summary, we've shown there exists both gut and skin dysbiosis in vitiligo patients. Although sampling skin surface is easy, it gives us limited information, and sampling deeper skin layers is more informative to study skin microbiota, at least for vitiligo patients. Using biopsies, we've been able to show that lesional skin has a unique microbiota compared to the other sites, and that dysbiosis in lesional skin is associated with depletion of protective bifidobacterium, enrichment of mitochondrial DNA, oxidative stress, and immune activation in the same patients. Whether these changes are a cause or a consequence remains to be confirmed. Nevertheless, it gives us some new therapeutic targets to examine for potential treatment of vitiligo. Finally, I would like to thank the entire Team 12, but in particular my talented PhD student Anen Bouget and our collaborator Professor Tina West from the UMEA University, who did the microbiome sequencing, as well as ISIS Pharma, who funded this project. I thank you for your attention.
Thank you very much, Dr. Meri, for a wonderful presentation. And we have a few questions for you. Is there any significant variability of diversity within the groups?
You mean regarding the patient characteristics?
No, indicate, just a minute.
Can you hear me? Yes?
Yeah, I can hear you.
Wonderful. The variation between...
I can restate the question. Sorry. I was just noting that there was diversity within the group, and your second to last slide actually suggested that. So you had the 3 patients who had no dysbiosis versus all the others within the same group with dysbiosis. Yes. And the question was whether there was a difference in activity there. I asked my question too soon, I think, because then you got to that slide that suggested that there was some difference there, which I think is really interesting.
Not a problem. Yes, those 3 patients. In fact, when we saw those such clear differences in their responses, we are very well aware that it's a very, very small sample size. And however, the responses are very clear. We saw a black and white response. And one of the obvious and the first explanations we started to look for is whether there was a difference in obvious characteristics such as patient age, disease status, also where the biopsy was taken from and things like that. None of these were possible explanations for the difference in responses, except, which is the reason why we actually went and looked at the immunity in these patients, because there's a lot of literature now starting to emerge showing a clear relationship between mitochondrial stress in vitiligo patients and potential relationship with particularly innate immunity.
Did you have a chance to compare the gut microbiome from the stool sample?
Yes, in fact, from these patients, as you can imagine, it was a quite invasive study where we obtained lots of different samples from each of the patients. We had, as I've mentioned, the swab, the biopsies from each of the lesional and non-lesional sites, as well as stool samples and blood samples. So we did look at stool samples and we did show that there is a significant increase in the FB ratio in vitiligo patients compared to healthy controls.
Is there any correlation with the age and dysbiosis?
Unfortunately, we don't have enough patients, certainly in this study, to be able to answer questions like that. But certainly in our cohort, we had a very, very similar age across the 2 groups. So certainly in our cohort, age doesn't explain at all the differences in responses.
And there's another question. How many swabs did you perform from each patient? Did you check dry and wet skin areas? And in the literature, there's an increased ratio of an autoimmune disease relationship thought to be causal. If so, what is the proposed mechanism?
Very good question. Regarding the samples, we sampled twice, a standard for the swabs, we sampled twice the same 5 centimeter square regions on the patients. And these swabs were done in a systematic manner. So exactly the same way for both of the groups. For the biopsies, we also performed the biopsies on the same body part. So for most of these patients, the biopsies were actually taken, for example, from the forearm, and all the samples were taken from the dry regions of the skin. Because we know, and it's well documented in the literature, there is a great biodiversity between dry and oily skin. So we always took from the dry areas and performed the experiments in a very systematic way to try and eliminate as much variability between these patients.
Yeah. Many of us are interested because from this particular basic study, what would be the therapeutic implications with regard to this particular study, because there's a lot more hope in this particular area.
Sure. This is just a very first study, and of course, these results need to be replicated in the larger cohort. But in theory, the clinical implications would be to actually replenish some of the crucial commensal bacteria we have shown to be missing on the surface of the skin of these vitiligo patients, such as it's done for many other conditions already in practice. So addition of probiotic, I guess, cocktails, or this is something that is for much later on, and the studies will need to be done, of course. But we have shown that we have certain strains of bacteria which are depleted, and these are commensal, such as Staphylococcus and bifidobacteria, which we know have really a potential and a beneficial effect with certainly immunity, and to be able to replenish the bacteria in the patients of these skins is something that we can envisage for future therapeutic interventions.
That's a very interesting discussion about the probio and the microbiome, and that it will not end. But we're out of time, and we'll have to shift for our next chair. So thank you all. Interest. Yes.
Thank you.
Yeah. Thank you very much.
Thank you.
Let's go to, we are going to close this particular session and over to Dr. Louis and Van Giel.